Welcome to EPG Pathshala. Students, in the previous module, we have discussed briefly about genetically modified organisms, their benefits, limitations, what are the different rules and regulations regarding the safety of genetic modified organisms, and what are the different regulatory authorities that deal with these genetically modified organisms, whether they are plants or animals. Now, coming to the introduction part, gene transfer is a process that involves the transfer of foreign gene into a host cell so that the desired product can be obtained. Now, this gene transfer can be obtained by sexual as well as vegetative production. However, it takes long time. Therefore, scientists are currently trying to use genetically engineering techniques for gene transfer in plants. The methods for the production of genetically engineered plants require the involvement of various vectors. Now, what is a vector? Vector is a round shaped DNA. The full form of DNA is deoxyribonucleic acid that has the capacity to independently exist as well as replicate. It acts as an artificial vehicle to carry genetic information from donor to receptor. Now, two types of vectors are used especially in case of plants. These vectors are Agrobacterium tumefaciens denoted by Ti and Agrobacterium rhizogenes denoted by Ri. Both Ti and Ri plasmids have almost same characters, like their tDNA region that can be transferred in any species and then integrated into genome. Vectors have some unique features like low molecular weight, genetic marker and unique restriction enzymes. There are different types of gene transfer processes in plants that are either vector mediated or vectorless. Now, coming to the next slide, what are the different vectors for plants? Generally, plasmid and viral genome based vectors, they are used for the plants. Plasmid based vectors. Now, coming to Ti plasmid based vectors, Ti plasmid is found in Agrobacterium tumefaciens. This strain is naturally occurring gram-negative soil bacteria. It is very important to note that it is a gram-negative soil bacteria. It has two species like Agrobacterium tumefaciens and second one is Agrobacterium rhizogenes. Both of these, they act as natural genetic engineers because they have the ability to transform the plants. Agrobacterium tumefaciens is responsible for crown gall disease in plants by inserting tDNA from plasmids with size up to 200 kb. Size of plasmid always vary in the range of 15 to 30 kb depending upon the strain. Coming to the next slide, what are the different genes that are required for the transfer of tDNA along with the mechanism? Now, Vir genes or virulence genes are the genes that are required for the transfer of tDNA into host cells. Vir genes, they are also known as virulence genes and contain Vir A, Vir B, Vir C, Vir D, Vir E, Vir G and Vir H genes. These genes are involved only and only in DNA transfer. They do not get transferred along with tDNA. Now, how the process of tDNA transfer is initiated? The process of tDNA transfer is initiated with the plant wounding. In this step, that is, during the wounding step, the bacteria are introduced into plant wound. Now, what happens next? Wounding leads to the production of some substances such as acetosyringone. Acetosyringone is identified by virulence gene. Now, Vir-A gene is a member kinase. It senses the phenolic compound and gets autophosphorylated on the histidine residue on the binding of acetylserine. Thereafter, what happens next? The phosphate group of Vir-A is transferred to aspartate residue of Vir-G protein. Now, it should be noted that Vir-A and Vir-G, they act together for the activation of other virulence genes. tDNA, that is transfer DNA, is nicked from Ti plasmid by Vir-D1 and Vir-D2. This is very important that Vir-D1 and Vir-D2, they are endonucleases. Next, Vir-D2 remains attached to the nicked tDNA at its 5' end. Nicked DNA, it gets placed from the plasmid and produces single-stranded tDNA. Now, single-stranded tDNA is susceptible to attack by nucleases. Therefore, to protect it from the attack of nucleases, Vir-E2 binds to that single-stranded tDNA. Next step is Vir-E2 helps in the transport of tDNA into the nucleus of plant cell. During the transfer of tDNA, energy is required, and that energy is provided by Vir-V11 protein. This Vir-V11 protein has ATPase activity. Vir-F is also involved in tDNA delivery. The process of tDNA transfer is a polar process that begins at right border and abolishes at left border. The single-stranded tDNA is thereafter converted into double-stranded DNA and gets incorporated inside the plant genome by a non-homologous recombination. This question often comes in competitive exams. Now, coming to the next slide, there are other methods that are direct, or we can say they are vectorless methods. Direct transfer of genes into receptor has been proved that it is simple and effective method for the transfer of foreign DNA. This method is further divided into 3 categories. These are mentioned below: physical gene transfer method and chemical gene transfer methods. Coming to the next slide, that is physical gene transfer methods. In this method, there is no natural vector required for the transfer of DNA to the receptor plant cell, and therefore, DNA is introduced directly without the help of any vehicle. This process is also referred to as DNA-mediated gene transfer. Now, various physical gene transfer methods, they are discussed in detail in the upcoming slides. The first physical transfer method is electroporation. It is the most popular physical method because of its simplicity, cheapness and fast nature. In this method, what happens? Electric field is applied so that DNA can be introduced inside the plant cells. Now, what happens? When electric pulse is applied across the membrane, small pores ranging from 40 to 120 nanometer, they are formed. Before the resealing of the pores, target material is introduced in plant cells. Some factors affect the efficiency of electroporations, and these factors are temperature, electric field strength, ionic concentration, DNA conformation and DNA concentration. Now, recently this technique is applicable in sugar beet and rice plants. Coming to the next slide, what are the different steps that are required during electroporation? First is harvest the cells during the logarithmic phase. Thereafter, centrifuge the sample at 40 degrees centigrade for 5 minutes. Resuspend the cells in growth medium at the concentration of 1:10 cells per ml. Thereafter, electric field is applied for 1 to 2 minutes and then introduce the electroplated cells to cultured dish and grow the cells. Next step is to analyze the DNA, RNA and culture cells continuously to get positive cell lines. This method has certain benefits as well as limitations. It is simple and very quick and also has low cell toxicity and inexpensive as compared to other methods. Moreover, small amount of DNA is required. However, this process is difficult to regenerate plants from the protoplast. Moreover, high intensity of pulse can destroy the protoplast. Some pores of larger size may be formed during electroporation process and the transformation protocol is laborious. Coming to the next physical transfer method, that is particle bombardment. This method was developed by Sanford et al. In this method, high density genetic material is transferred with high velocity so that DNA can be introduced into the receptor plant cell. What happens during the process? That gene gun shoots a piece of DNA into the recipient plant cell. This DNA piece is coated with tungsten or gold and fired across the stopping screen accelerated by helium into plant cell. This method is applied for the wounding of the plants so as to increase the agrobacterium transformations. This method is also known by other names also like particle acceleration, particle inflow gun or gene gun. This method has also certain advantages and disadvantages. Coming to the advantage part, bombardment process, the binary vector is not required in this process. It is a simple protocol and is applicable on all plant species. However, it has certain disadvantages like equipments are very costly and during the transformation process there are chances of DNA damage. Therefore, it leads to non-homologous integration of chromosomes. There would be a difficulty in getting single copy of transgenic events. Third method of physical transfer is macroinjection. It is a very simple method. In this method, DNA solution is injected into developing plant parts and those plant parts can be shoots. And how the DNA solution is injected, it is done with the help of micropipettes. The injection volume of genetic material lies between 5 to 10 microliter. Next method is microinjection. This is also a simple method. In this method, DNA is introduced directly into the target cells under microscopic control. Now, this method has a very unique property that it can even penetrate the intact cell walls. However, in this method, the cells have to be immobilized first. Now, how the immobilization can be done? Immobilization can be done by agarose embedding, agar embedding, suction holding pipettes and polylysine treated glass surface methods. This method has been applied to raise the transgenic plants of tobacco and Brassica napus. This method has certain disadvantages. It is a very slow process. Sometimes chimeric plants or false plants can be produced. It is an expensive method and highly skilled and experienced staff is required. Next method is liposome-mediated transformation. This method employs the fusion of protoplast using PEG. PEG is polyethylene glycol. DNA enters into the protoplast by endocytosis of liposomes. This technique is useful in tobacco and carrot. The process involves some steps such as liposome firstly get adhered to the surface of the protoplast. Thereafter, liposomes get adhered at the site of adhesion and finally, the plasmids get released inside the cells. The advantages of this method are it has low level of cell toxicity, there are less or little chances of DNA damage. It is applicable on wide range of cells, highly reproducible, stable and safe storage can be done. Next method is silicon carbide fiber mediated transformation. This method employs the use of silicon carbide fibers that have great intrinsic hardness with sharp edges. The process involves the vortexing of silicon carbide fibers, plasmid DNA and tissue in the Eppendorf tube. The fibers have 10 to 80 micrometer length and 0.6 micrometer diameter. During the mixing process, DNA gets penetrated into the plant cells through silicon carbide fiber. This technique has been used to raise genetic plants in maize and tobacco plants. The benefits and limitations of this method are that it is a simple procedure and is affordable. Moreover, few types of equipment are required. The only disadvantage is that silicon carbide is carcinogenic in nature. Next method is ultrasound-mediated DNA transformation. This method is also known as sonoporation. In this technique, plasmid DNA and plant cells, they are mixed together with buffer in sonicator. Ultrasonic pulses, they are applied at 0.5 watt per centimeter square intensity for 30 minutes. Thereafter, the samples are rinsed with buffer and are cultured for further growth and differentiation. The example of this method has been used in tobacco plant. There are the benefits and limitation of this method. It is inexpensive, multifunctional and it is easy to standardize. Sometimes high intensity of ultrasonic pulse, they can cause damage to the cells. Till now, I have discussed only physical gene transfer methods. Now coming to the chemical gene transfer methods. These methods, they involve destabilization of plasma membrane with chemicals. Plant cells are incubated with foreign DNA in buffers having poly-L-ornithine, polyvinyl alcohol and polyethylene glycol. Polyethylene glycol mediated gene transfer. For the first time, it was tested on petunia and tobacco in presence of polyethylene glycol and poly-L-ornithine. Steps involved are: firstly, protoplasts are isolated, plasmid DNA is mixed with protoplast suspension and to this mixture 40% of polyethylene glycol is added. This solution should be added slowly since it has high viscosity. The mixture is incubated for 5 minutes and thereafter the efficiency of PEG method is affected by magnesium concentration and calcium ions in presence of donor DNA during the incubation period. The advantage of this method is that it is a completely controlled experiment and intermediate vectors are not required. The only disadvantage is that protoplast is required. Next chemical method is calcium phosphate co-precipitation method. This method involves the mixing of donor DNA with calcium chloride and isotopic phosphate buffer so as to form DNA calcium phosphate precipitates. The precipitates thus formed react for several hours with actively dividing cells. The precipitates are then washed and incubated in culture medium. DMSO, dimethyl sulfoxide, enhances the transformation efficiency and the success of transformation depends upon DNA concentration. Next method is the polycation DMSO technique. Polycation and polybrine are used to increase the surface area for the adsorption of foreign DNA. Thereafter, 25% DMSO treatment is given to accelerate membrane permeability and increase the uptake of DNA. This method is advantageous since it has less toxicity as compared to the other polycations and moreover small quantities of plasmid DNA are required. Coming to the next method, that is DEAE dextran procedure. This method involves the use of diethylaminoethyl dextran. This chemical forms a complex with DNA and then causes the transformation of plant cells. DMSO shock increases the efficiency of transformation up to 80%, but all the transformations, they are not stable. Coming to the next chemical method, that is virus-mediated gene transfer. Some viruses have been explored for their potential to be used as vectors. Three vectors that are commonly used are colimoviruses, Gemini viruses and RNA viruses. Out of these, RNA viruses do not have the potential to be used as a vector. Now coming to the application part. Now first is insecticidal resistance. Major reason behind the reduction in crop production is insect attack. To handle this problem, some insect resistant plants, they have been developed on the basis of the insecticidal proteins. During the sporulation process, Bacillus thuringiensis, that is the bacteria, it produces alpha endotoxins, Cry1, Cry2, Cry3 and other proteins. These Cry proteins, they are toxic in nature. After the ingestion of endotoxins, these proteins, they bind with the midcut of the epithelial cells and cause the damage of the epithelial surface. This is an effective method to control the insects in various crops such as cotton, tobacco, tomato, potato, maize, rice and canola. Second application is the production of virus resistant plants. Virus resistant plants, they have been developed through several approaches including ribosome-mediated technology, introduction of coat protein gene and antisense RNA technology. The best approach amongst these is the introduction of coat protein gene. First transgenic virus resistant plant was tobacco that was produced in 1986 containing tobacco mosaic virus CP gene. After tobacco, many other plants, they have been produced using this technology such as papaya and watermelon. Uncoating of virus particles is a necessary process and is required for the expression and replication of viral genome. However, CP gene blocks this process and provides a resistance against virus. Next application is production of herbicidal resistant plants. Weeds are unwanted plants that grow with the cultivated plants and compete with them for the nutrients and space. When herbicides, they are applied for weed killing, the crops also get affected. The herbicides inhibit the process of photosynthesis in crop plants. However, this process, that is photosynthesis, is an essential process. Therefore, some crops have been engineered genetically for herbicide resistance. Let us take an example of glyphosate. Glyphosate is a non-selective herbicide and is a competitive inhibitor of 5-enol pyruvyl shikimate 3-phosphate synthase, that is EPSP. This is a very important enzyme that is required for the synthesis of aromatic amino acids. Aromatic amino acids include phenylalanine, tyrosine and tryptophan in plants. Shikimate and phosphophenol pyruvate, they are converted into EPSP. Therefore, two approaches have been used to produce resistance against the glyphosate herbicide. First approach leads to the overproduction of EPSP. Now, overproduction will provide resistance as number of enzymes will be available to the cell. This technique has been used in tobacco, tomato, soybean, canola, sugar beet and cotton. In the second approach, mutant version of EPSP synthase gets expressed. This mutant version possesses resistance to the herbicide. Now, next application is the modification of plant nutritional content. Genetic engineering plays a crucial role in the improvement of the nutritional value of crops. Applying this process, lipids, vitamins, amino acids and iron content can be increased. The famous example is the production of vitamin A enriched golden rice. In rice, vitamin A is very low. However, Potricus and Bayer, they developed a new variety of rice that is known as golden rice by a genetic engineering technique. This variety is enriched with provitamin A. For the development of golden rice, 3 genes, namely phytoin synthase and lycopene synthase, they were introduced. These 3 genes, they are involved in the biosynthesis of carotenoids. The carotenoids are the precursors of vitamin A. Next application is use of plants as bioreactors. Transgenic plants, they can be used for the production of antibodies, that is plantibodies, chemicals and proteins on the commercial scale, since they can grow easily and produce large biomass. Nowadays, biopharmaceutical sector is trying to eliminate animal derived proteins by plantibodies, since there are more chances of contamination of animal derived proteins and that can prove pathogenic. Second is edible vaccines. Production and packaging of commercial vaccines is very expensive. Therefore, moreover, trained persons, they are required to administer the injections. The solution of this problem is the production of edible vaccines as fruit or vegetable. The edible vaccines do not require elaborated packaging, sterilization, purification and delivery. Suitable parts of plants, they are consumed by humans to enhance the immunity of body. Examples of edible vaccine plants include potatoes, bananas, tomatoes, lettuce, carrot, etc. Next application is delayed ripening. Softening of fruits due to ripening process is a major problem as it causes economic losses. The process of ripening can be delayed using genetic engineering approach. Basically, what we know that polygalacturonase is an enzyme that is responsible for the fruit ripening. This enzyme acts on the polygalacturonic acid of the cell wall and results in gradual softening of fruit. Expression of polygalacturonase enzyme has been reduced via antisense technology. With the help of this technology, a permanent copy of antisense for the PG gene has been introduced using Agrobacterium tumefaciens. In this technology, antisense RNA binds with opposite sense mRNA and forms a stable duplex. This duplex prevents the formation of normal amount of polygalacturonase and interferes with the process of translation. This technique has been demonstratedly effectively in tomato. Next application is the development of stress resistant plants. There are several types of stress such as low water availability, flooding, high salt concentrations, high and low temperature that are having negative effects on the growth and production of plants. Metabolic genetic engineering has been used for the synthesis of salt and cold tolerant rice variety. Now, what are the advantages of genetically modified crops? They have better flavor and high nutritional value. Productivity is high as well as yield is also high. Certain unwanted characters have been eliminated. GM crops, they act as vaccine carrier. For example, banana. They show delayed ripening and can be stored for longer period. Virus and herbicide resistant plants have been developed. Growth of GM crops is faster in comparison to the other crops, and they have the high capacity to tolerate stress. Moreover, they are also helpful in the conservation of water, soil and energy, and lastly, GM crops are eco-friendly. This is the major advantage. Now coming to the disadvantages of GM crops. GM plants, they decrease the diversity if they are grown for longer period. Cross-pollination of GM plants with other plants bring unwanted changes in plants, producing negative effects on quality as well as on quantity. Sometimes transferred genes, they have some negative effects on human body by causing allergies. Pollens of GM plants have some undesired properties such as respiratory and skin diseases. Then comes environmental problems. Gene pollution is transfer of gene from GM plants to weeds and pests to make them superweed and super pests. Leakage of GM proteins in soil causes soil pollution and proves to be dangerous for soil flora and fauna. GM plants are harmful for natural pollinators like insects and butterflies. For example, BT corn produces pollen that are highly toxic to monarch larvae and black swallowtail. GM proteins are responsible for the surface and groundwater pollution. Students, at last, I would like to conclude the module by saying that we have discussed about the various vectors, whether they are physical vectors, physical methods or chemical methods required for the formation of genetically modified organisms. Thank you.